Good morning. Let's discuss on the metabolism of phenylalanine and tyrosine. They belong to the group of aromatic amino acid, and the only benefit of phenylalanine is to convert up into the tyrosine. So in this diagram, we'll see how the phenylalanine is going to be converted into tyrosine. Phenylalanine requires the enzyme phenylalanine hydroxylase, which is dependent on the presence of on the supply of oxygen and tetrahydrobiopterin as a coenzyme. So in course of this conversion, the oxygen, one atom of the oxygen is added to the water molecule, while the another oxygen is added onto the phenylalanine phenyl ring, and it's added as an hydroxy group of the tyrosine. So tetrahydrobiopterin is converted to dihydrobiopterin in course of this conversion, and dihydrobiopterin which further reverts back to tetrahydrobiopterin in the presence of enzyme dihydrobiopterin reductase, which also requires the presence of NADPH. So if any of the enzyme is lacking, like phenyl hydroxylase or the this dihydrobiopterin reductase is deficient, so it may be like it may lead to the total blockage of this path, and there is no conversion of phenylalanine to tyrosine. So that will result into the severe accumulation of phenylalanine, and which can further converted up into the various other metabolites, unrequired metabolites via alternate route, and leading to the disease that is known as phenylketonuria. Now we'll see in the diagram, like phenylalanine is converting up into the tyrosine, and rest of the catabolist steps are there of tyrosine. How tyrosine is going to convert to various intermediates via different enzymes. So ultimately, what is the end product? All we'll see in this slide. So from phenylalanine to tyrosine, which we have seen in the previous slide, the enzyme is para-hydroxy phenylalanine hydroxylase. Further, tyrosine in the presence of enzyme tyrosine transaminase converts up into the corresponding keto acid, that is the para-hydroxy phenyl pyruvic acid, which requires the presence of alpha ketoglutarate, and PLP is required, that is uh, um, important coenzyme for the reaction. I already discussed in a separate video about the transaminase reactions and how they are facilitated by PLP. Then further, para-hydroxyphenyl pyruvic acid is converting up to dihydroxyphenyl acetic acid, that is homogentisic acid, which requires the enzyme para-hydroxyphenyl pyruvate hydroxylase, and this is dependent on the presence of oxygen, which further converts to carbon dioxide, and ascorbic acid and copper 2 plus ions are required for the enzyme to facilitate the reaction. For if this enzyme is lacking, para-hydroxyphenyl pyruvate hydroxylase deficiency will lead to the tyrosinemia type 2. We'll discuss later on this. That is also known as neonatal tyrosinemia. Then further, dihydroxyphenyl acetic acid, that is the homogentisic acid, that is going to convert to mallyl acetoacetic acid in the presence of enzyme homogentisic acid oxidase, which requires also the presence of ferrous 2 plus ions, and oxygen is required for this conversion. So if the enzyme is lacking, this enzyme is deficient, this homogentisic acid oxidase, that will lead to the metabolic disorder, that is alkaptonuria, and which leads to the lot of accumulation of homogentisic acid in the blood, which is accumulated into the various bones and cartilages also. Urine is black color. Urine is a very diagnostic feature of this disease. Then mallyl acetoacetic acid is converting to fumarate acetoacetic acid in the presence of enzyme mallyl acetoacetate isomerase. Further, fumarate acetoacetic acid is cleaved up into two components in the presence of enzyme fumarate acetoacetate hydroxylase. So two components are created or are produced. The one is the acetoacetic acid, and second one is the fumaric acid. And we know the fumaric acid is an important intermediate of Krebs cycle, and all intermediates of Krebs cycles can undergo gluconeogenesis and leading to the formation of glucose. That's how it is. It is apparent that phenylalanine and tyrosine are glucogenic as well as ketogenic because they are generating acetoacetic acid also as the end product, and acetoacetic acid to acetyl coenzyme, which is the precursor for synthesis of long chain fatty acids and ketone bodies. So that's how it is. Both ketogen these both amino acids are ketogenic as well as glucogenic. So have a look on the metabolic disorders associated with phenylalanine and tyrosine. So phenylketonuria I have already discussed in detail. There is a separate video on the phenylalanine disorder, that is phenylketonuria. So in this is associated with inability to convert to phenylalanine into tyrosine. Alternative pathway of phenylalanine to catabolism in phenylketonurics, because phenylalanine is accumulated. So instead of converting to tyrosine, it is converting to various other metabolites in the form of phenylacetate, in the form of phenylacetylglutamine, phenyllactates, and phenylpyruvates. And so they can accumulate at various sites of the body and leading to various clinical features. So for phenylketonuria, you can consult my video on uh, phenylketonuria. Then tyrosinemia type 1. So this is due to the deficiency of fumarate acetoacetate hydrolase or melyl acetoacetate isomerase enzyme deficiency. It results in accumulation and excretion of tyrosine and its metabolites. So these are the various metabolites of tyrosine: para-hydroxyphenylpyruvate, para-hydroxyphenylactate, para-hydroxyphenylacetate, N-acetyltyrosine, and tyramine. So clinical features are like liver failure, kidney dysfunction, polyneuropathy, and vitamin D resistance rickets. This diagram shows the alternative pathway of tyrosine to various other metabolite. Via tyrosine transaminase, it converts to para-hydroxyphenylpyruvate, then from L-tyrosine to N-acetyltyrosine, then tyrosine decarboxylase lead to tyramine, then tyramine converts to para-hydroxyphenylacetate in the presence of enzyme tyramine oxidase. Then dehydrogenase will lead to conversion of para-hydroxyphenylpyruvate to para-hydroxyphenylactate. So this is how the various metabolites are produced. So the clinical features associated with diarrhea, vomiting, cabbage-like odor to the skin and urine is due to succinyl acetone. It is an abnormal metabolite which is produced via fumarate acetoacetate. So death may occur in infancy due to the acute liver failure. Diagnosis can be diagnosis can be done via detecting these tyrosine metabolites and succinyl acetones in urine. Then molecular genetic testing for FAS, that is fumarate acetoacetate hydrolase gene mutations. Then newborn screening is to measure the succinyl acetone via tandem mass spectroscopy in the blood and urine spot samples. Then carrier testing and prenatal diagnosis by DNA analysis are also available if the specific gene causing mutations has been identified in the family. So prenatal diagnosis is also possible by detection of succinyl acetone in the amniotic fluid. So treatment based on low phenylalanine diet and tyrosine diet is recommended. Then second type of tyrosinemia is type 2. That is also known as Richner-Hanhart syndrome, and this is due to the deficiency of tyrosine transaminase enzyme deficiency. So Then clinical features are associated with the accumulation of tyrosine and its toxic metabolites in the blood and tissues. I already showed in the previous slide how the alternative pathway lead to the various other metabolites, unexpected metabolites. So they are accumulating, these toxic metabolites are accumulating in the blood and tissues and appear in the urine. So accumulation of tyrosine causes the eye lesion, dermatitis, and mental retardation. So diet low in tyrosine and phenylalanine is recommended. Diet with vitamin C may benefit the corneal and skin lesions of tyrosine aminotransferase deficiency, but it cannot cure the mental retardation. Then third type of tyrosinemia is type 3, that is also known as neonatal tyrosinemia. Defective enzyme is para-hydroxyphenyl pyruvate hydroxylase. So serum tyrosine level are high in premature infants, resulting in from an immature liver and its limited ability to synthesize the enzyme para-hydroxyphenyl pyruvate hydroxylase. That's how there is serum level of tyrosine is very high. So as the liver matures, the accumulated tyrosine is metabolized and the serum level decreases within 4 to 8 weeks of age. It is a benign condition and it responds well to the ascorbic acid because the enzyme, concerned enzyme, sometimes it is not active, so gets activated via the coenzyme, that is ascorbic acid.